Good afternoon, everyone. Welcome to the H3D Foundation webinar series. Our today's speaker is Dr. John Woodland. Dr. Woodland works as a research officer at the Holistic Drug Discovery and Development Center, H3D, at University of Cape Town. Dr. Woodland is interested in developing molecular strategies such as treatments and tools to tackle the troubles of infectious diseases. His interdisciplinary research experience spans the fields of chemistry and biochemistry. He is also passionate about promoting science and improving scientific literacy in South Africa. So today's topic is Bright New World, Harnessing Fluorescence for Exquisite Insights into Chemical and Biological Systems, a crash course. And now I would like to request Dr. Woodland to share his work with us. Over to you, Dr. Woodland.
Thanks so much, Shruti. Really nice to be here. Thanks everyone for joining in today. So as Shruti mentioned, this is going to be a crash course, so we're going to move super quickly, but I hope it's just going to give you an overview into fluorescence, why it's a really cool technique and how we can use it to learn a lot about chemical and biological systems. So just to start with something to whet your appetite a little bit. Here is a CHO cell that's been illuminated with GFP-linked microtubulin, and GAP-DH mRNA has been stained, and so is the nucleus, which is very typical in biological imaging. Here's another cell. This is a super resolved image of a T cell, which has now been stained with various markers, including to illuminate some important proteins in the HIV virus. So the GP120 protein has been labeled. And then you'll notice those blurry yellow spots are diffraction limited GAG proteins. So that's a protein for which a super resolution image couldn't be acquired. You can see how it's a lot more pixelated relative to the rest of the cell. And here are some GFP labeled mycobacteria. It's a cytosolic expression of that green fluorescent protein, but it's tagged at the surface with a purple antibody. And just to thank my colleague at UCT, Karen, for sharing these images with me. So a bit of an outline today. I'm just going to take you through the basic photophysics. What is fluorescence? And what are some of the terms we associate with fluorescence? What do they mean? Things like spectra, brightness, photobleaching. Talk about the different types of fluorophores, dyes, fluorescent proteins, quantum dots. And then we'll talk a bit about fluorescence spectroscopy and some of its applications. And then move on to, I guess, the main course, if you like, fluorescence microscopy, and some common applications of that technique. And then we'll talk a bit about quantitative image analysis and some considerations for bioimaging. I know that's why some of you are here today. So really just hoping in this webinar to give you an introduction to fluorescence and how we can sort of harness it for generating interesting insights into chemical and biological systems. And I hope that after this, you'll be inspired to consider incorporating fluorescence into your own work. Towards the end, I will share some recommended resources and also my details. So you're very, open to contacting me later if you'd like to chat about any of these things in a bit more detail. Perhaps you're wondering where the title of today's talk came from, Bright New World. So it's a bit of a play on Brave New World, which is a fantastic dystopian novel by Aldous Huxley. But he took the title of his novel, Brave New World, actually from a play by Shakespeare called The Tempest. And in that play, Miranda is one of the characters. She's been brought up on an island with her father. So she has not been exposed to other people in her life. And one day there's a shipwreck. And suddenly all these crew wash up and she is just dazzled by all of these people. She can't believe that so many beautiful creatures exist in the world. And so that's where Huxley took the title of his novel from. And so I thought, you know, in an analogous way, the way Miranda was so dazzled by all of these people in the world, so fluorescence has helped scientists to really uncover all this complexity and detail in biological systems we didn't know existed before these technologies developed over the last few decades. So yeah, here are some of the topics we're going to be chatting about today. But why fluorescence, you may be asking? I mean, we have a lot of great chemical stains, but they are limited in terms of what they can do. They often require fixing the cells in specific ways, and they're limited in terms of their sensitivity and their specificity. We do need to stain our samples though, because if you just look at cells under a microscope, there isn't terribly much detail there. But if you look at a fluorescence image, for example, you can see how we really get great signal and a very specific signal as well. So some of the advantages of using fluorescence over other imaging techniques is that you get very high contrast, we get very good specificity and sensitivity, very good stability. If you prepare your samples in a fixed way, they can last for many months, if not years. Importantly, the technique is quantitative as well, which is really valuable for a lot of biological system imaging work. And you can use live cells as well, which tends to introduce fewer artifacts into the imaging analysis. So just a few words of history, because many of you probably enjoy drinking gin and tonics. And as Shruti mentioned, I'm interested in infectious disease and malaria in particular. So quinine is a very important molecule. It's a constituent of tonic water and is a very effective anti-malarial in many contexts. And perhaps the scientists were doing some daytime drinking about 200 years ago. But John Herschel was in fact the first scientist to record an observation of a quinine solution in sunlight. He noticed that this solution had an extremely vivid and beautiful celestial blue color. And it's also just interesting to note on the side that John Herschel actually spent a lot of time here in Cape Town. So in a way, Cape Town and South Africa are also kind of linked up with the history of fluorescence in some respects. But a few decades later, Herschel's colleague, George Stokes, actually coined the term fluorescence. And in the same way that opalescence, which is another optical effect, comes from the name of a mineral, he thought that this phenomenon that we sometimes see in some minerals, and you'll see here is fluorspar or fluorite, that also fluoresces. So he decided to call this phenomenon fluorescence because this particular mineral has fluorine in it. Fluorine actually has nothing to do with fluorescence. It's actually some lanthanide contaminants in the mineral that actually give the fluorescence. And so it's kind of funny in the history of science how these names are often sort of incorrectly given to things, but then persist over time. So fluorescence, contrary to what you might think, doesn't specifically have anything to do with the element fluorine, for example, but it's the name we use today. And since then, there've been this incredible 150 years of development in this technique where really now we can image, single proteins inside cells with exquisite precision. So what is fluorescence? It's a photophysical process, and at its simplest is where light is absorbed by a molecule and then light of lower energy is released. So here's an example of a molecule called fluorescein, and when we excite it with light in about the 480 nanometer region, we get fluorescence of a particular color. In this case, it's green. It's a two-step process. First thing that happens is there's absorption of a photon that then excites an electron to a higher energy level. And then when that electron returns to the ground state, there's the release of energy as a photon, as a lower energy photon. So fluorescence is on the one hand an absorption and excitation process, and then to actually see the photon that is released afterwards requires relaxation and then the emission of that energy as light rather than as heat, for example. And specific wavelengths are required for fluorescence. So if we illuminate something like fluorescein at too long a wavelength, we don't actually see the light because that energy does not correspond to the correct gap in energy of those molecular orbitals. And we use the Jablonski diagrams to represent these electronic states within a molecule. So you have the ground state, and in this case, the first excited state. These are singlet states. I won't go into the electronic details too much, but essentially you get absorption of a photon, a loss of some energy as heat, often then dissipated as kind of vibrational energy. And then when you get the electron returning to the ground state, that energy is released in the form of a photon. And that's what we call fluorescence. Now, this can happen at multiple wavelengths, at some longer wavelengths where less energy is released than was absorbed. And you can see that you get a whole manifold of energy levels that then reveal themselves either at the fluorescence stage or at the absorption stage. These correspond to a variety of different wavelengths. And so that's why when we look at the spectra of these molecules, either their absorption or fluorescence spectra, excitation or emission spectra, they tend to have these envelope shapes. They're not these very narrow bands, although some fluorophores do have those. Just to talk a little more detail about, because you've heard me mention the word fluorophore now, that's distinct from chromophores, which are really just any groups in molecular structures that have the capacity to absorb light of particular wavelengths. And these correspond then to energy differences between the molecular orbitals, as we saw in the Jablonski diagram. And excess energy can be transferred or given off as heat, but a specific type of chromophore, a fluorophore, is one that emits light upon excitation. So rather than just dissipating that incoming or absorbed energy as heat, it's emitted as light. And these molecules typically contain these aromatic or planar or cyclic groups with pi bonds, because we need these delocalized, these conjugated systems in order to give the stabilization of that excited state that allows for emission of a photon, which manifests as fluorescence. So larger structures, as you may remember from basic chemistry, with longer conjugated systems, usually result in longer fluorescent wavelengths, so more into the green or the red region, as opposed to the blue, which will be the shorter wavelength or higher energy. And these molecules typically are quite rigid because they're aromatic, and so their pi orbitals kind of lock into place. And typically these are small molecules, what we might call dyes, but you can even get fluorescent proteins, fluorescent nanoparticles that we'll chat about in a bit. And a very famous fluorescent probe, besides the quinine that I mentioned a bit earlier, is fluorescein, which was first synthesized about 150 years ago. And that really kick-started all these explorations into fluorescence. So just a few more words about excitation and emission spectra. They tend to look like this across your electromagnetic spectrum, the visible region, and the gap between the maxima of those excitation and emission peaks is what we call the Stokes shift. And it's important that this is an appropriate value. It's not too long and it's not too short, and we'll chat about that in a little bit. But essentially, all fluorophores then can be characterized according to their excitation maxima, their emission maxima. And these days, there are thousands of fluorophores or fluorescent proteins really spanning all the way from the blue into the red. And they all have different advantages and disadvantages. As is often the case, there isn't a one-size-fits-all, but some fluorophores are more ideal for particular applications than others. One very important criterion of a fluorophore is what we call brightness. So this is kind of a measure of, if you illuminate the fluorophore, how much light do you want to get out the other side? And of course, we generally like things that are bright because we get a better signal to noise ratio. But if you illuminate your fluorescein, say here, with 10 photons, do you get 10 photons out? Well, not necessarily, because some of those photons might not be absorbed in the first place. And even if those 8 remaining photons are absorbed, some of those might not manifest as fluorescence. And we call these two properties the extinction or the molar coefficient on one hand, and the quantum yield on the other. So the extinction coefficient measures how readily a chromophore, more generally, or specifically here, a fluorophore absorbs light. So something with a higher extinction coefficient absorbs light more easily or more readily. And then we also look at the quantum yield, and something with a higher quantum yield is more efficient at converting those absorbed photons into light. So once again, a higher quantum yield is preferable. And we can equate these two properties into a value we call brightness, which is essentially just the product of the extinction coefficient and the quantum yield. And some people divide it by 1,000 to make the number a little bit easier to read. But you can see there how these fluorophores then all have characteristic extinction coefficients and quantum yields. And if you put those together, an idea of brightness, bright generally being better. There's a dark side to fluorescence as well, so one also gets non-radiative decay. So not all of those excited electrons will return to the ground state and release light. Some of them will just dissipate as heat. You get non-radiative decay where heat comes off instead of light. And we typically call this quenching. And depending on the application, that can be good, but usually it's undesirable. Sometimes, though, one gets what is called intersystem crossing, where the excited ground state electron, or rather the excited singlet state electron, moves to a triplet excited state. This is not ideal because it can then be, it's in a very reactive state because one has unpaired electrons in the excited triplet state, and this can react with molecular oxygen and cause a permanent loss in fluorescence. This can also be a good thing sometimes because it actually gives rise to phosphorescence, which is a related and in some ways very useful technique. But as a fluorescence scientist, this intersystem crossing to the excited triplet state, which is highly reactive, the molecule can then react with molecular oxygen, it can lose its fluorescence. That's usually undesirable. Fortunately, this is quantum mechanically, statistically very unlikely, but it does still happen when we're talking about gazillions of molecules. So the general term for this effect is photobleaching, when a fluorophore loses its fluorescence due to this damage, which is often oxidative damage induced by light. And you can see here how it manifests in a biological situation where you have a cell and it's now been photobleached, and this is a permanent loss in fluorescence, which can be advantageous in some respects, but generally speaking is not ideal. Some of you who've done fluorescence imaging before will know that there are steps we take to avoid this photobleaching, things like staining and keeping the samples in semi-dark conditions, really reducing the excitation or exposure time when imaging, and trying to reduce that laser power as much as possible. Of course, the brighter a fluorophore is, generally the less energy you need to get a good signal on the other side, which is why we like bright things. I should just mention, although this is a topic for a whole separate talk, a really valuable technique is called FRET, or fluorescence resonance energy transfer. And this is a distant dependent process in which we get a non-radiative energy transfer from one fluorophore to another. So an excited donor can essentially give its excited electron to another fluorophore, the so-called acceptor, via an intermolecular long-range dipole-dipole coupling. So I know this all sounds quite complex, and it is, but in a biological context, it's quite easy to harness this to really tell us useful things. For example, whether two proteins are localized in space. There are certain criteria that have to be fulfilled. One needs a spectral overlap of these two fluorophores. One needs them to be in the correct orientation and within a close enough distance to one another. But just mentioning to you that this is a very important technique that has been well exploited in biological imaging. And in fact, they call it a spectroscopic ruler because you can sometimes use it to measure very small distances within the cell. So just to wrap up this first section, a bit of a quiz. What are some of the properties of an ideal fluorophore? So I mentioned that the molar absorbance or extinction coefficient, generally higher is better because that means your fluorophore is more likely to absorb light. And generally a higher quantum yield is better because it's more efficiently able to convert that absorbed energy into light as opposed to releasing it non-radiatively as heat. The excitation and emission wavelengths of the ideal fluorophore will depend on your application of interest. So whether that you're interested in imaging something in the blue region of the cell or the red region of the cell will depend on your co-dyes, any autofluorescence, other things going on. And this will be closely related to the Stokes shift as well. You don't want a Stokes shift that's too small, otherwise you can get a lot of spillover or bleed between the excitation and emission, but it can also be tricky if the Stokes shift is too large. That can also have problematic consequences. Generally, you'd like a fluorophore that is more photostable. This is a bit of a trick. Fluorescence lifetime is something we haven't spoken about yet, but is also very important in fluorescence studies. And that's the amount of time the electron stays in the excited state before it comes back to the ground state and releases the energy as light. So again, we need an optimal fluorescent lifetime, not too short, but also not too long, because if the electron is too long in the excited state, there are other quenching processes that could cause deactivation of that electron before we get the manifestation of fluorescence. And depending on your application, whether you're doing live cell imaging or whatever the case might be, things like solubility, permeability, reactivity are going to be important as well. So we're going to change sort of direction a bit now, just to talk about different types of fluorophores and to give you a sense of scale between small molecules, larger molecules like fluorescent proteins, and then big nanoparticles like quantum dots. So the most ubiquitous fluorophores tend to be small molecule dyes, and you'll probably recognize some of these names, the rhodamines, the coumarins, the alexofluors, some of which tend to be very bright and photostable. And a nice thing about these fluorophores is they're generally quite sensitive to their environment. So you can tell useful things about ionic concentration, pH, temperature, that kind of thing. Lots of advantages. They're often bright, photostable, they're commercially available. Some of the disadvantages, though, can be that they require covalent linking or fixing in a biological context, and one needs to be quite careful that some of this environmental sensitivity, which, while it may be a good thing, can also come back to bite you and cause some problems. But there's a whole range of them, and a lot of people have done surveys, and you get great charts like this that really show what some of the most bright fluorophores are across the wavelength range, for example. So you can really sort of pick the fluorophore that best suits your desired application. Then we have the fluorescent proteins that were discovered some decades ago from this jellyfish here, the blue luminescent protein, and its partner, the green fluorescent protein. And Roger Chen in particular, who won the Nobel Prize, did so much work to really explore and understand how these fluorescent proteins work. So typically, one expresses or one links the gene for this green fluorescent protein to the gene for your protein of interest. So when the cellular machinery then translates your protein of interest, it also transcribes and translates the GFP, which essentially is a tag or a marker or a reporter to then tell us where your protein of interest is in the cell, which can be great for monitoring expression or localization of proteins within cells. And you get a vast range of these fluorescent proteins, red fluorescent protein, yellow fluorescent protein, with a whole bunch of more exotic names as well. I'll show you in a sec. But one does need to be careful, because having a large protein tagged to your protein of interest can cause a lot of structural perturbation. It can influence its expression, its localization. It can even influence its activity. So you do need to do a lot of validation just to show that when I've linked my fluorescent protein to my protein of interest, the protein of interest is still behaving the way you want it to, so that any observations you then do, tell you something useful about the system and are not just an artifact now of your engineered system. And as I mentioned, you get fluorescent proteins in a whole host of different colors with all sorts of different photophysical properties. And then quantum dots, these are particularly useful in a materials context, so developing maybe better diagnostics or better technologies like screens. They can be very, very useful for those kinds of things. Again, with several advantages: very narrow absorption and emission peaks, very bright. They're not susceptible to photobleaching in the way that small molecule dyes are covalently susceptible to photobleaching. But they have disadvantages as well, especially in a biological context. They're very large size. Their toxicity means that they're not often used in bioimaging studies. How then does one attach small molecules, particularly small molecule fluorophores or dyes, to biomolecules? Well, generally, chemical or covalent modification is the way we'll do that. That's a bit more tricky, but this is really where things like click chemistry and bioorthogonal chemistry come into their own, especially if you can do those chemical reactions in situ, actually, within the cellular milieu while you're doing the experiments. But you can also use genetic means with fluorescent proteins, as I showed you a few slides ago. And immunolabeling still remains very effective to this day, where antibodies have fluorophores attached to them, although typically one does need to fix these cells. And so one can't do live imaging studies necessarily. And then for nucleic acid studies, fluorescence in situ hybridization, some of you may know FISH, is now a ubiquitous technique and is really fantastic at looking for particular strands of nucleic acids within a cellular environment. Here's just a pretty picture as a commercial break. So now we're going to move on to spectroscopy and microscopy. So here's just a reminder of the electromagnetic wavelength and how in the visible light spectrum, red light generally has a longer wavelength, it's lower energy. Blue light moving into the ultraviolet region around 400 nanometers has a shorter wavelength and hence is higher energy. And you're probably all familiar with absorption spectroscopy, how a spectrophotometer works. One can do fantastic studies to show binding and all kinds of things. And this is also a quantitative technique. We know of the Beer-Lambert law. And so when one does fluorescence spectroscopy using a fluorometer or a spectropluorimeter, essentially it's just like absorption spectroscopy, but on steroids. And so a spectropluorimeter typically will look like this, where you have a light source. That light is then going to enter an excitation monochromator, and this is very useful because you can define exactly what wavelengths of light you want to use to excite your sample. The fluorescence is then emitted and goes through an emission monochromator, so we select just the wavelengths we want to read, and then those go to the photomultiplier tube where they are enhanced for the detector to make sense of. And you'll notice here, importantly, the fluorescence is coming off the sample at right angles because we don't want the incoming light or the exciting light to be picked up by the detector. Because remember, you need to excite with a particular wavelength of light, which you specify in a spectropluorimeter, and then you want to read it at a particular wavelength or range of wavelengths. And you don't want any interference between that incoming light and the fluorescence that is emitted by your sample. And one can do all kinds of fantastic experiments in the lab. Generally, it's valuable to characterize any fluorescent materials you make, fluorophores or fluorescent conjugates, and this is what one of those spectra will look like. So you see on the left-hand side, we generally like to show the excitation or absorption spectrum on the one hand. On the right-hand side, the fluorescence emission spectrum, and then of course wavelength along the axis at the bottom. I've also indicated the Stokes shift. This is once again quinine, in case you're wondering what its excitation and emission profiles look like. So you can see once again how it's active in the UV. We need to excite it at about 330, 350 or 60 nanometers. Ultraviolet light you get in the sun outside, and that's when you see this blue fluorescence emerging at about 450 nanometers or so. So this is quite a good guideline to use if you want to present your fluorescence spectra in a particular way. But remember, it's always important to specify exactly the conditions in which you acquired these spectra, the concentration, the solvent, the temperature, because as we saw a little bit earlier, especially small molecule fluorophores are very susceptible to small changes in chemical environment. This can be harnessed for good, of course. If you've made a fluorescent drug conjugate, say, you might want to look at the effect of solvent on the fluorescence. So if you're asking the question, how does the fluorescence profile of my compound change in different environments? You can then look at a whole bunch of different solvents and look at the fluorescence maximum or the fluorescence intensity in those solvents. And then you can extend this to a biological situation and say, well, if I know that, let's say, membranous compartments, lipids in my cell, have polarity values that are about the same as these solvents, then in the cellular system, I can look for a fluorescence wavelength or a fluorescence intensity that corresponds to the kinds of readings I'm getting in these solvents. So this is how you can build a correspondence between a chemical and a biological situation. But you can imagine how even these simple cuvette-based experiments can be extremely valuable in characterizing new fluorophores or new conjugates. In the same way, we can use absorbance spectroscopy to tell us a lot about binding. We can also look, and we do this frequently at H3D, at metal ion chelation with spectropluorimetry. So the question, does my compound bind or chelate to a physiologically relevant metal ion like iron, nickel, cobalt, whatever the case might be? We can do things like Job plots. You can then monitor the fluorescence if your compound's fluorescent. That's the caveat here. Although this technique tends to be extremely sensitive. And in this particular case, we don't see any binding, but where we see a deviation from linearity in the Job plot, that is suggestive of, in this case, quite strong binding, which gives us a good hunch that this particular molecule we're looking at is actually interacting, in this case, with iron 2 metal ions. But now we come to the big gun, to the fluorescence microscope. It's a very complex piece of equipment, as you can see, and I'm not going to go through all the important components, but there are things like light sources, and generally we use lasers in some of our more advanced confocal microscopes, collimators, polarizers, just to make sure that all the light is correctly in phase and is monochromatic, filters to make sure that appropriate wavelengths are permitted to pass through or are excluded, beam splitters, and then detectors as well. We need typically very sensitive detectors because we might only be seeing just a few photons of fluorescence coming off the sample. But just something important to mention is resolution and contrast. And as many of you will know, resolution really is just the amount of detail in an image, what we think of as like the quality of the image. And that's very important. If you're doing a cellular imaging study, you need an appropriate level of resolution, particularly if your cells are very, very small. And what's also going to be bound up with resolution is contrast, is making sure you have enough signal to noise. And that's one of the reasons we love fluorescence as a technique, is generally our background, our noise, if you like, is very, very low, which means any fluorescent signal we're getting is really quite pronounced against that background. So this is what the simplest wide field microscope looks like, really not too different from that spectrophlorimeter we saw a few slides ago, where you have your light source, a lens, and an excitation filter letting only certain wavelengths through, a mirror reflecting some of those photons down to the sample, and then the sample is going to emit fluorescence if all goes according to plan, and those photons are then going to be permitted through the emission filter focused by the lens to the detector. So the simplest wide field, what we call an epifluorescence microscope, is very versatile. Be careful though, generally you don't get any axial information. So just like this picture can give you the wrong impression. So if you have very thick samples, you might also think that some organelles are maybe lying in close proximity to one another when actually they're separated through space in what we call the Z-axis. So perhaps the most ubiquitous solution to this is what we call confocal microscopy, where you have a movable stage that can move through various Z depths or heights. And then we have a pinhole that excludes any out of focus light. And so this means we don't have a wide field or epifluorescence situation, but we're only looking at light from a very thin slice of the specimen. So we know that anything lying in this plane is from that plane and is not from above or below. And that can be very useful. Can also have its disadvantages though. It means because the pinhole is excluding a lot of out of focus light, you often need to irradiate your sample with a lot more energy, a lot higher power from the laser, which can sometimes be damaging. So as with everything, it's a balance, and you've got to decide as the experimentalist what your priority is and what's most important. There are a few variations on this theme. One also gets what's called TIRF, or total internal reflection fluorescence microscopy. And this can be very useful for thicker specimens to look at exactly what is on the cover slip, what is the closest to the lens there. And that's wonderful for measuring, say, cytoskeletal dynamics. One can also use light sheet microscopy, or so-called selective plane illumination microscopy. And this is wonderful for in vivo imaging, or even imaging whole organisms like zebrafish or mice. And this allows this volumetric imaging of often live samples in a very gentle way with minimal light exposure and hence minimal light damage. And again, a topic for another time, but really one of the most exciting developments in microscopy over the past 20 or 30 years, and what won the Nobel Prize almost a decade ago, is what we call super resolution fluorescence microscopy. So this is techniques that now break this diffraction barrier of light, where up to a certain point you can't get any better detail, you can't improve the resolution. These scientists and others have come up with ways and techniques to enable us to break that diffraction limit. And we're very lucky here in South Africa in particular, we have many super resolution instruments that are able to exploit these techniques in order to generate really unprecedented insights into cells. So what are some of the applications then, coming to the last part of this talk, of cell imaging? Well, many laboratories use fluorescence to measure cell viability and cytotoxicity. It can also be great for measuring things in real-time functional assays. So looking at calcium flux, for example, can be is one type of experiment that has really done very, very well in this fluorescence modality. One might want to look at cell migration or movement over time, perhaps changes in morphology before and after treatment of a drug, say, drug localization studies, subcellular studies in real time, whether it's autophagy or endocytosis or protein trafficking. And then much more what we call complex experiments, things like the FRET I mentioned a bit earlier, if maybe you want to look at the relationship between two fluorescently labeled proteins. Or another it's not so complex, it's a fairly simple experiment, but it's sort of thought of as a more complex experiment, is what we call FRAP, or fluorescence recovery after photobleaching. So what you do there is, in your cellular environment, you actually deliberately and intentionally photobleach, very strongly irradiate a portion of the cell, and then you wait and you see what fluorescent molecules diffuse back into that cellular compartment or whatever it is that you're looking at. And so that fluorescence recovery after photobleaching, FRAP, can also be very valuable at looking at those kinds of dynamic events within a cell. If you're going to look at fluorescent conjugates or drugs under a fluorescence microscope, and you're going to synthetically make these molecules, you do need to make sure you characterize them carefully. So just a few tips. Obviously, you need your structural information, NMR, LCMS, if you've attached, let's say, a fluorescent dye to a drug-like molecule because you want to look at where it's going in the cell. It's generally good to get an absorption profile of that compound. 5 micromolar in ethanol is normally a good starting point, and then use that same sample to get the fluorescence profile. And you might at this point want to interrogate different solvents, see how your fluorescent drug conjugate, how its brightness, how its photophysical properties are changing according to different solvents. And then you also probably want to evaluate its solubility, and importantly, any binding data, especially any biochemical or biological evaluation you can do. And this is so crucial because once you've now introduced a fluorophore onto your molecule of interest, it could change the physicochemical properties of that molecule quite significantly. And so you need to make a good case that whatever localization studies or imaging experiments you're going to do with this fluorescent drug conjugate are in fact a good surrogate, a good mimic, a good substitute of the parent compound. We've done some of this work here at UCT, and this is just to show you in the context of malaria parasites, Plasmodium falciparum. We did some super resolution imaging and how you can really tell some cool things, in this case, a fluorescently labeled chloroquine molecule, and where that localized relative to other cellular structures. The second last part of this talk, and I did warn you, sorry, there's going to be a crash course, a whistle-stop tour, really just to make you aware of all these things because they are important. I don't think we spend enough time talking about quantitative image analysis. Too often people are very happy just to put a representative image in a presentation, in a publication, and just assume that's okay. But then I'm always reminded of this fantastic advice from almost 300 years ago. Beware of determining and declaring your opinion suddenly on any subject, for imagination often gets the start of judgment and makes people believe they see things which better observations will convince them could not be seen. Therefore, assert nothing till after repeated experiments and examinations in all lights and in all positions, when you employ a microscope, shake off all prejudice, nor harbor any favorite opinion. So, perhaps good advice for us all as experimental scientists to remember. And there are lots of different tools one can use to evaluate images and look at things like colocalization. There's the cytofluorogram. There are these fantastic correlation coefficients, Pearsons, Manders, and really great tools like ImageJ that can help you to get these numbers. And then when you have a case like red-green colocalization, are my signals, my channels overlapping? It looks like they do, but can I show that in a quantitative way? then you can really get the numbers that will then back up these suspicions you may have. And in the same sense, when things don't co-localize, it's important to show quantitatively that that's not the case. And it just makes any observations from your microscopy far more representative. It reduces bias because you're necessarily having to do this over a very large sample size, but it's avoiding the subjectivity that might come into the microscopy. It's not all plain sailing. There can be a lot of problems, issues, complications with the fluorescent probes. The photobleaching I mentioned earlier, this irreversible covalent damage to your probes. Quenching can often be a problem as well, especially in particular cellular environments or self-quenching. So we just need to be careful of that. Saturating the detector can happen very easily. Sometimes molecules have very broad spectra, and so that can create big problems when it comes to isolating your photons and making sure that the signal I'm getting is just from my probe or my dye and not from other things. And then toxicity and autofluorescence, particularly in plant samples, can be a big issue. So one just needs to do all the appropriate control experiments to make sure that you've got all your bases covered. Where to with new probes? Well, we're always on the lookout as bioimaging scientists for wider ranges of wavelengths, especially in the red and infrared region. These then are probes that require much lower energy for excitation and so are much more suitable for live cell imaging. And up till now, people haven't really explored that part of the electromagnetic spectrum. We always want probes that have better solubility, are brighter, or have increased photostability. And these days also, for super resolution techniques, special properties, things like photo switching, and we're getting a really new, exciting series of fluorescent reporters coming into mainstream use, what we call snap tags or clip tags. So these have all the benefits of genetic reporting, genetic encoding, but with small molecules rather than large and bulky proteins. So watch this space. You may want to print screen or capture this slide. It's a really nice selection of resources that I think are a really good first port of call for anyone who's keen to do some imaging or spectroscopy work, or just understand a little bit more about fluorescence, super resolution imaging, and all the photophysics behind what's going on. I'd say these are the top things to read in the field. What facilities are there? Well, for those in the Western Cape, we've got great microscope over at the Molecular and Cell Biology building on Upper Campus at UCT, and some great microscopes on the Health Sciences campus as well, and also at Stellenbosch University, administered by their central analytical facilities. For those a little further afield, the South African bioimaging community was launched last year and is really growing. That's an exciting space where people are sharing protocols and publications, so we don't need to reinvent the wheel. The University of Cape Town also hosts AMI, the Africa Microscopy Initiative, and they frequently have calls where you can then apply to do your imaging experiments using these state-of-the-art instruments. And more broadly speaking, on our continent, if you are based outside of South Africa especially, please consider joining our African Bioimaging Consortium, or ABIC, which is a really valuable community for imaging scientists, once again, to share notes, provide advice. Imaging can sometimes be, in fact, as you can see here at my microscope, can be quite lonely sometimes, many hours spent in the dark. And so it's always nice to know that you have others who you can turn to for advice and moral support. So yes, thank you for your attention. Just a final note that while we've seen a lot of pictures in this presentation, the purpose of microscopy is insight, not images. So it's interpreting those images in a useful way that can then help us to generate new insights into chemical, into biological systems. So thanks very much for your attention. And as I mentioned earlier, please drop me a line if you're interested in knowing more, or if you'd like to discuss how maybe fluorescence can be applied to your projects. My email address is over there.